Hello, friends. Good afternoon. Am I audible to everybody? Is my voice coming properly to you guys? Great. So we have crossed one hurdle. That is the hurdle of voice, right? Great. So we'll be starting exactly in 5 minutes. And I hope you liked yesterday's session. Today we are going to discuss about the cell culture first. Then we'll be taking a break of 5 to 10 minutes. And then we will be discussing about another technique which is called as real-time PCR. So that is today's agenda. So I hope we'll be having a good day today. If you are having any question from yesterday, you can ask right now. I can give some answers in next 5-6 minutes till we start. So guys, we are starting in next 2 minutes. So guys, yesterday on day one, we have discussed mainly about the flow cytometry, different fundamental principles of flow cytometry. Then we have discussed some of the good applications, some easy applications, some little bit difficult applications, and so many things. Today, our focus will be discussing the animal cell culture specifically. And generally, when you learn animal cell culture, then people learn about different type of cell cultures, different type of media, cell culture media, decontamination techniques, safety, and all these things. But I am not going to discuss these things. I am going to tell you some of the very important things which you have to remember in cell culture if you really want to be a good cell culture person. Something new, something with a different approach towards the cell culture. So I'm going to start this PowerPoint presentation, and if you are able to see the slide, then please give your response here. If all of you are able to see the slides on your screen, then I will be starting. So I will be sharing the PowerPoint presentation on the screen. Then I will be taking a break of 60 seconds. And if you are seeing it, then we will start. Okay. So guys, are you able to see this PowerPoint presentation? Great. So the PPT is visible to all of you guys. So let's start discussing about world of cell culture and actually what we should know about it, right? So I start this presentation, this discussion with a amazing line which I firmly believe in my life, that humans are selfish, so are the cells. Okay, and in next 1 hour, you will be having a different point of view of thinking towards cell culture experiments, and actually towards the complete biology. So humans are selfish, so are the cells. What do you mean by that? So humans always ask selfish questions, always do selfish things, and they do the same practice of asking selfish questions and selfish things. They do the same practice in biological research also. I give you one very simple example. If I ask somebody a question that, what do you think why the cells are dividing, or why the cell division is important? Okay, then most of the people give the answer that cell division is important for growth, it is important for body formation, as well as it is important for repair of the organs. Now, at surface level, this answer looks fine, but I am having a problem with this answer. And the answer is, the problem is, this is a selfish answer. Growth is happening for whom? Humans. Repair is happening for whom? Human organs. Whole body is forming for whom? Humans. So we as humans are giving answers from our perspective. If I ask the question why the lactobacillus bacteria is producing lactic acid, when in that step where you convert pyruvate to lactic acid, there is no generation of energy, then suddenly we think that we are getting lactic acid. Yeast is making alcohol for us. Some other bacteria is making ethanol, methanol, butanol for us. Similarly, we think that a plant is making starch to give us potatoes. Similarly, we think that cells are dividing to give us our body, to give us our organs, to grow us. Similarly, we think a protein is folding because the folded proteins are functional. If you look closely to all these statements, you will find that all these answers and all these statements are made from the perspective of humans. So I call them as selfish approaches towards biology. And if you really want to be a good biology student and a good biology researcher, then what you have to do, you have to change your perspective. You have to change your point of view. You have to start thinking not from your own angle, but from the perspective of the system itself. Why proteins are dividing? I will be thinking from the point of view of chemical structure of protein. Then I will get the answer. If I ask a protein, why are you dividing, sir? Why are you folding, sir? Then proteins will say, my friend, when we are in a primary structure, there are so many amino acids which are hydrophobic in nature, and we are present in the cytoplasm, which is containing huge amount of water, and it is our moral duty to protect these hydrophobic amino acids from surrounding water. And the best strategy to do that is to fold in such a way that our hydrophobic brothers go inside and our hydrophilic brothers come on the surface. Then I am getting the right answer. Proteins are folding to protect the hydrophobic amino acids from water of cytoplasm, the globular protein. And the resultant of this process is what? They are functional. They are not getting folded because they want to be functional. They are getting folded because they want to stabilize their structure. If I ask the cells, ma'am, why are you dividing? Then cell will say, bro, we eat a lot, we do good amount of metabolism, and because of that we quickly falls into a problem. The problem is the volume of the cytoplasm is going to be increased definitely, but the volume of nucleus is not getting increased. So after some time the nucleus starts losing the control on cytoplasm, and to best way to re-establish that control is to decrease the size and change the volumetric ratio again, and the best method to do this to divide. And then we ask the cells, ma'am, okay, that is your reason of cell division, then you tell us how do you know that this problem is happening, and now you have to start the division. Then cells will say, we get the external signals from nearby cells which can tell us now it is time to divide, now it is time to grow, now it is time to increase your number. So we just sense the environment, we just sense our own issues. That's why we divide. We don't give a damn about you humans. And if you really want to culture us, then understand our requirements. And if you understand our requirements, then you will be the master of what? Cell culture. You will be having a great point of view towards cellular experiments, and you will become a very amazing biology student. So guys, from today itself, we'll try to give the answer of every biological question or every biological problem, not from our perspective, but from the perspective of what? Cells, proteins, or you can say from the perspective of system itself. Then we'll not have to remember anything. We'll start getting amazing ideas. We will start getting amazing results, and we will be learning every concept of biology in an easier and in the most efficient way. So in next 1 hour, when I will be discussing the cell culture, then we will be doing only one thing. We will be learning the requirements of what? Cells, different type of cells. And then we will be saying, if we really want to culture them, then we have to do this, we have to do that. I know that I can teach pipetting to an art student in 5 days. What do you have to do? You just have to tell him, press 1, press twice, suck it, release it. That's it. For that, you don't have to be a student of biology. That is the work of a technician. So you are a student of biology. So what you have to do? You have to design the experiment. You have to analyze the experiment. You have to make your own protocols. You have to understand others' protocols. You are not there to do only pipetting. Pipetting is a daily job. The more important thing is learning the real fundamentals of cell culture. Right? Contamination will obviously come. First time, second time, third time, you cannot stop it. But you can stop it by doing what? Practice. Good pipetting, practice. Sterilization, practice. Right? But if I tell you, from tomorrow you have to start the culture of B lymphocytes, you cannot do practice for that. Practice can be done with the technique. But to start the culture of B lymphocyte, what you have to do? You have to learn about B cells. You have to learn about cells requirements. That is why before starting this topic of world of cell culture, I told you this line: humans are selfish, so are the cells. Okay? Great. So we start learning one by one different basic fundamentals. Then I will be giving you information about some definitions and everything. So we can learn a lot by understanding a developmental pathway. Okay. So a developmental pathway in terms of cellular division and cellular differentiation tells us this. The life starts from embryonic stem cells. Then from embryonic stem cells, we get progenitors of adult stem cells. Then from the progenitors of adult stem cells, we get adult stem cells. Then from adult stem cells, we get what? Differentiated cells. Right? So when I go from through this flow or through this path of embryonic stem cell to differentiated cells, two things are happening. First is their differentiation potential decreasing. Means the embryonic stem cell can make all types of cells of our body. Progenitors of the adult stem cells can make some of the cells. Then adult stem cells can make only one or two types of cells, and differentiated cells cannot differentiate further. Similarly, cell division potential is also decreasing. So you can say the fully differentiated cells like keratinocytes, they cannot divide. So generally the cell division happens in our body during the development of embryo in uterus. These cells are dividing, embryonic stem cells and these cells. And after that, once we are adult, then whenever we require the cell division, then these adult stem cells will divide. Okay, differentiated cells will not divide. So how the system is achieving that? Right. I can give one very nice example of that. When we cut our hair, then what happens actually? Cutting hair means you are removing some cells. So you require the repair. And to do repair, you have to divide the cells. Now that cell division is not occurring in the hair cell. That cell division is occurring into hair stem cells, which is the example of what? Adult stem cell. So when a hair stem cell divides, then it gives you two types of cells. One, the hair stem cell or adult stem cells again, and second, the differentiated hair cell. So what you are having, you are getting a new hair cell as well as you are maintaining the population of the adult stem cell, because if you cut your hair next time, the same thing can happen. Third time, same thing is happening. Fourth time, same thing is happening. When I am having an injury to skin, and that injury has to be repaired by doing cell division, then the cell division does not occur in the external layer of skin because it is made up of only differentiated cells. The cell division occurs at the base of skin where there are skin stem cells. And these stem cells, they divide, they maintain their population, as well as they give you new cells. Imagine every day we are eating different type of food, and this food is reaching, and these complex materials are reaching to your intestine, and because of that rough food and different type of food, we lose many intestinal epithelial cells every day, every minute, every hour. You have to replace them. How you are replacing? By doing cell division into intestinal epithelial cells? No. Because intestinal epithelial cells are the examples of differentiated cells and they don't divide. What is happening? Intestine is also having intestinal stem cells. What intestinal stem cells are doing? They will do division. They will give you intestinal epithelial cells for repair, as well as they will maintain their own population. So in our body right now it is happening. Every organ or every tissue system is having a pool of what? Adult stem cell. And whenever you require the cell division, they can take charge, they will divide, they will give you a differentiated cell, as well as they will maintain their own population. That is how the cell division is occurring in our body. So this kind of cell division is called as asymmetric cell division, where a stem cell or adult stem cell is dividing and it is giving you one stem cell and one differentiated cell. So asymmetric cell division means two daughter cells coming from one mother cell, and one cell is similar to mother cell and the second cell is not similar to mother cell. Then these stem cells, to maintain their population, they can do symmetric division also. One stem cell can give you what? Two stem cells. So stem cells are having the property of both symmetric division as well as asymmetric division. By symmetric division, they are mainly maintaining their own population. So if you require the repair again and again and again and again, they can do it. And by asymmetric division, they are maintaining their own population as well as they are giving you new differentiated cell. But the crux is the differentiated cells are not going to divide. They can divide actually if they are having some mutations. By those mutations, they regain the properties of cell division, and these cases are generally cancer, right? So this is the developmental pathway by which we are learning which cells can divide, which cells cannot divide. You can pressurize the differentiated cells also, but they will become what? Cancerous. They will transform. They will lose some of their own properties. Now, we are having some definitions like primary cell culture, cell lines, or cell clones. Okay. If somebody asks me what is a primary cell culture, then I say you take out the tissue, you crush it, and you prepare the single cell suspension out of it, and you start growing them. But after some time they will die because they are having a finite lifespan. They don't have infinite lifespan. And this type of cell culture, which is directly coming from the tissue, is called as primary cell culture. Now, what do you mean by cell line? It's very easy to define a cell line. A cell line is permanently established cell culture, which is having infinite growth, and that will proliferate indefinitely given appropriate fresh food and space again and again. So you take one culture plate or culture flask, you put some media, they will divide, their number will increase. Then you divide them into two flasks, new feed, new media, new food, new space, then 4 flasks, then 8 flasks. You can maintain them what? For infinite time. Now, we are again having this pathway where the embryonic stem cell is giving you progenitor of the adult stem cell, then differentiated cell, and two properties are decreasing: differentiation potential as well as cell division potential. So the established cell lines we are actually getting from the primary cell culture only, and these primary cell culture on repetitive cultures can give you what? A established cell line, and these cell lines become immortalized and they get the ability to proliferate indefinitely. Now, here we are having an issue. What is the issue? That the primary cell culture is having a finite lifespan. Means after 7 days, 8 days, 15 days, it will die. Okay. But if you establish a cell line out of a primary cell culture, it means you are converting them into an immortalized system which is able to proliferate indefinitely. And how you can do that? By giving them cancerous properties like random mutations, like expression of telomerase, like insertion of cancer genes. And so what you are telling the cell lines, what you are telling a primary cell culture is, sir, you cannot grow more than 10 to 15 days. But I want to do experiments for 6 months, 12 months, 18 months. And for that I require what? Cell line. Right? And how I am going to make the cell line out of you, primary culture? By giving some of your cells the ability to be immortal. And who is immortal actually in the natural environment? The cancer. The cancer cell can grow for a infinite time, right? And I can make you immortal by giving you some mutations, by pressurizing you to again and again to be cultured, by expressing the telomerase, by inserting the cancer antigen. After that, you will be able to grow for infinite time, but with a little drawback. The drawback is you will get cancerous properties, right? That's why whatever cell lines we hear, which are available in the market, which can be grown for a very long time in the culture system, they are generally cancerous. HeLa, cervical cancer; 293T, cancerous; A549, lung carcinoma; ALC, again a cancer having cancerous property; CHO cell line, cancerous property; hybridoma mixed with myeloma. So it is very simple to say, it is very convenient to say that when we are growing the cell lines for an infinite time period, actually we are growing some cells which are having some cancerous properties. That is why if you go to PubMed and you start looking the research papers, you will find that with time, the impact factor of research papers based on cell line experiments is decreasing slowly. And the reason is very simple. People have identified that all these experiments which we are doing on the cell lines, they are not perfect. Why they are not perfect? Because the cells which we are using are having some cancerous properties. So they can give us some biased results, as well as they can give us the results which we don't want. That is why the system of biological research is moving towards mainly primary cell culture, and it is moving towards the animal models. So people take tissue, prepare the cells, culture them for a small time period, do their experiment, and whenever they want to do experiment again, they again crush the tissue, prepare the cell culture. So the cell line work is slowly decreasing. But we have learned a very important thing from the system and from the pre-established cell lines is that the cell lines are having cancerous properties, and that can be little bit problematic in different type of what experimentations, right? Now we move to our next point, how we are doing the culture, primary cell culture. Very simple. Here is the flow chart where you are starting the cell culture from mouse. You can dissect it. You can cut out the spleen, for example, or any organ. Then you crush it mechanically, which I have discussed yesterday. Then you can do enzymatic treatment of collagenase, displase, elastase to break the tissue and to free the cells. Then in these single cells, you add the media and you culture them for a small time period. This is your what? The generation of a primary cell culture. Now, similarly, what people have done, they have taken the biopsy, they have taken out the human patient tumor of melanoma, they prepared the single cell suspension by all these methods, mechanical digestion, then adding the enzymes and all these things. Then they established what? They established the primary cell culture, and then they pressurized the primary cell culture by different methods, again and again doing subculturing. And they have created what? The cell line. And we are having now so many cell lines which are getting used for gene expression studies, then immunohistochemistry studies, then people are testing their drugs and all. So it's a very simple method. If you ask a tissue how I get a single cell suspension from you, so then tissue will say my cells are attached to each other by different metrics and proteins like collagen and then elastin and all these. How you are going to get the single cells out of me? Very simple. You digest those proteins. So how I am going to digest the collagen? By collagenase enzyme. How I am going to digest the protein elastin? By utilizing the elastase enzyme. So cells themselves are telling us we are having these properties, and if you want to culture them, then do this. So that's why whenever I do cell culture in my lab and whenever I try to establish a cell culture based experiment, what I do, I ask the cells themselves. Asking them means I go for the literature, how they divide, how they live in their natural habitat, and can I copy that natural habitat in my cell culture flask? Then the answer comes, yes, you can take these growth factors, these proteins, you have to digest these proteins, you have to keep them like this. I learned everything from their natural habitat. And once I am doing that, then I am having a best protocol. Because I am fulfilling the requirement of the cells. I am telling them, okay, don't take tension. You have come out of your natural environment. I have digested you. I got you as a single cell suspension. But my culture flask is your new home. I am going to add all the signaling molecules, all the growth factors which you require. And then you will be growing happily with me and we'll be doing some nice experiments together. Respecting the cells, talking to the cells is important key of what? Doing cell culture. Now there are some other definitions. Now, it is a very interesting thing I'm going to introduce, that all these definitions we can understand and we can remember permanently if we are actually understanding the cells. First, there are two types of cell culture media: serum free versus serum containing media. Now, you are starting a cell culture and you are giving them sugar, you are giving them media and everything, carbohydrate source, everything. Then, if you are growing the hepatocyte or if you are growing a particular type of cells, and for cell division, they require what? External signals, they require growth factors. And if you don't have any idea about these growth factors, then what you will be doing? You will be adding the serum in what? Media. What is serum? Serum is cocktail of growth factor and signaling molecules which can tell the cells, okay, I am here, you divide. Sometimes we exactly know that what kind of growth factors and signaling molecules are required for a particular cell type. Then we don't add the serum. We add those molecules and those growth factors and those signaling molecules one by one. So in that case, we don't require the serum. We can make the media serum free by adding one molecule at a time. Then there is adherent cells and suspension culture. Again, very simple. The cells which are present in the solid tissue in our body, obviously they require the help of matrix. So they cannot grow in the liquid media, they cannot grow in the suspension. So obviously the liver cells will be growing into what? By sticking to the culture plate or by sticking to the culture flask. Reason? In the body they require the support. Melanoma cells require the support. Oral cancer cells require the support. Lung cancer cells require the support. T cells don't require the support because they are present in the blood. They can grow into what? Suspension culture. B cells can grow into suspension culture. Dendritic cells require support, so they will be growing out what? As adherent culture. Macrophages, adherent culture. Monocyte, suspension culture. Do you really remember all these definitions? Yes, if you are reading cell culture as a subject. But if you are reading cell culture as an extension of the cell biology, an extension of the fact that what are the natural requirements of the cells, then you don't have to remember them. Now, Some people are saying there are two types of method. So in one flask, the cells are grown enough, the flask is confluent enough, and you have to make two flasks out of one flask. Then either you can use trypsin enzyme, we call this process as trypsinization, or you don't want to use, or you don't have to use the trypsin enzyme, so there is no trypsinization. Do we have to remember it? No. If a cell is growing in adherent system, means it is sticking to your wall of your plate, or it is sticking to bottom of your plate, or it is sticking to your culture flask, then obviously how they are sticking? By their surface proteins. If you want to remove them and to divide them into two parts, what you have to do? You have to remove those proteins. What is the best method to remove those surface proteins? By digesting them. How we can digest them? By utilizing a protease enzyme. Which protease enzyme is generally being used in the cell culture? Trypsin. If a cell is growing into suspension culture like T cells, B cells, and if you want to divide them into two parts, are you going to use trypsin? Not at all. Reason is simple. They are in suspension. They are not sticking in onto the walls, and if they are not sticking, they are not having those surface proteins by which they can stick. So you don't need the digestion of those proteins. You just shake it and divide it into two parts. Easy to understand trypsinization or no trypsinization. Then sometimes cells require support of nearby neighboring cells. Then we can give them neighboring cells in form of what? Feeder cells. And some cells don't require the help of neighboring cells. You ask the cells, do you require the help? They will say, go for the literature, and in the literature you will find, okay, this kind of cells is not requiring any help of neighboring cells or any other cell layers to divide. Then we don't require what? Feeder cells. All these requirements and all these definitions are not there to remember. These are the experiments which you actually learn by asking the right question to the system itself, and by looking at the answer of the right question from the perspective of system itself. The day when you become selfish, you cannot do biological research experimentation. Then you start having biased point of view or biased thought process towards the experiment. Now we discuss some complex things. Okay, this I have already discussed yesterday, that different type of cells in cell culture can be recognized by their cellular phenotype, and this phenotyping we do by utilizing the CD proteins, like the cluster of differentiation proteins. We have used also CD3 yesterday, which was the marker of T lymphocyte, CD19, which was the marker of B lymphocyte, and these are so many different molecules which can be utilized to identify the cells and whether you are growing the correct cell or not. Right, you have taken out the blood. From blood you are taking out the PBMCs, and from PBMCs you are trying to grow the T cells. Okay, and if you are trying to grow the T cells, then what if there is some contamination of B cells also? What you will be doing to check the contamination, to check the quality of your cell culture? Very simple. You add these two antibodies, and in flow cytometry, if you are getting some percentage of B cells, then you will be saying my culture is contaminated with other type of cell. Otherwise, if you are having 95, 99, 100% of CD3 positive cells, then you will be saying, okay, my cell culture is what? Good quality. I am growing the right type of cell. And this quality checking you can do time to time by utilizing different CD markers and flow cytometry, or some other techniques are available also, like morphology, and then checking some transcription factors, real-time PCR and all. So now we see some complex examples that what we should learn to culture some particular type of cells, and what we have to learn and how we have to apply those learning in specialized cell culture. And one of the important topic here is stem cell niche. Very important. Now, what do you mean by the word niche? So niche we actually learn in ecology, and Niche is actually defined in ecology as a territory, or in common words or in simple words, I can say niche is nothing but the comfort zone. Comfort zone means an area where you are most comfortable. And if you come out of that area, you start behaving oddly, or you start behaving like something else. Okay. So what are the stem cells? Stem cells means the cells which can give you new cells, as well as the cells which can maintain their own population. And as I have told you, that stem cells are doing all the functions related to repair, growth, and all these things. Okay. Now, what will be a stem cell niche? Stem cell niche will be an area, a 3D space where stem cell will behave as stem cell. And if it comes out of that, then it will be losing its stem cell properties. Right? And how we define that area? That area is having particular cell type. That area is having particular signaling molecules, combination of signaling molecules, which are telling the stem cells that we are making a nice environment for you. And if you stay here, then you will be always stem cell. And if you leave this area, then you will not be getting proper signals, and then you will not be behaving as stem cells, and you will be converting into something else. This example of hair fiber and related system is very nice. So at this position, we are having what? Bulge stem cells. They are the type of what? Stem cells. If they go downwards, so this is the stem cell niche. In this area, they are behaving as bulge stem cell. Now, if they go downwards towards the root, then they start giving new cells of hair. If they go in this direction, by leaving their niche, they start making sebaceous gland. And if they are going upwards, they start giving you other type of cells. Same cell is giving you two types of cell depending upon what is their external environment. Now, if I want to grow the bulge stem cells in my culture flask, what I have to do? I have to learn what are the properties of this area, what kind of signaling molecules they are having, what kind of growth factors they are having. Now, after culturing bulge stem cells for some time, I want to convert them into sebaceous gland cells. Then what I have to learn? That I have to stop all the cell signaling molecules and growth factors which were coming from here, which I was giving initially, and I replace them with the growth factors and signaling molecules which are present in this environment. I want to convert them into these cells, then I have to change the environment of my culture media. I have to put what? New growth factor sets and new chemical signals or new cell signaling signals. I can grow the stem cells. I can differentiate them also in vitro in my culture plate or in my culture flask just by changing their niche, just by changing their micro environment. One more example we see. So imagine this is our bone. This black line is the wall of bone. This is second wall of the bone, and inside the bone we have bone marrow, and we are having HSC, hematopoietic stem cells. So the hematopoietic stem cells, which are into the contact of osteoblast, or you can say the bone matrix cells, they remain as what? Hematopoietic stem cells. But after division, if they go away, so this cell is dividing into two parts, two cells. One is still in the contact of what? Osteoblasts, and one is away from that contact. It will be differentiating into something else. That is how hematopoietic stem cells can give you different type of cells. If the cell is leaving the environment of bone marrow and going into thymus, then it will start converting into what? T cells. Now somebody asks me, can you convert hematopoietic stem cells into T cells in your system? Yes, we can try. will be giving step by step all the signals which they are naturally getting first in the bone marrow and then into what? Thymus. And if I replicate that system, then I can grow, or actually I can do the in vitro differentiation, or I can convert the hematopoietic stem cell into any type of target cell. So this area near to the bone, near to the wall of the bone, is the niche for what? Hematopoietic stem cell. Now, practical example I give you. People grow embryonic stem cell in their lab. People grow induced pluripotent stem cells. They are actually similar to embryonic stem cells in their lab. But when they do it, one of the biggest problem they face is they start the culture with embryonic stem cells and they start the culture with induced pluripotent stem cells. But these stem cells quickly convert after 2 or 3 days into something else. So it's a challenge to maintain the population of what? Embryonic stem cell or induced pluripotent stem cell in our culture system or in our lab. What people have done? They have identified a protein or a signaling molecule which is very important to maintain the niche. Maintaining the niche means if that protein is available in the culture media, then these stem cells will remain as stem cells. They will not convert into something else. And if you remove this protein from the culture media, they will start differentiating into something else. And the name of protein is LIF1, LIF1, leukemia inducing factor 1. And if you start the culture with induced pluripotent stem cells or iPSCs, and you put LIF1 into your culture media, then they will remain as iPSC for a very long time. And a day you start thinking, okay, now it is enough. I am having good number of iPSC. Now I want to convert them into something else. I want to convert them into some another type of cell. What you do? You remove the LIF and you replace the LIF with the protein specific to the target cell. It is everything about learning the niche and learning the microenvironment, learning the growth factors, signaling molecules which are required by different types of what? Cells. And once you do that, you are you are efficient, you are the master of what? Cell culture experiments. And you can achieve that by, again, changing your point of view, asking the right question, and giving and searching the answer from the perspective of the system itself. And why I am repeating this line again and again? Because I really want everybody of you to become a good researcher. And you will feel with time, if you really want to achieve your goals, then the most important thing right now is changing the point of view, asking the question and giving the answer from the perspective of the system itself. Don't be selfish, right? And you already know, if you become selfish in your life, then there are so many negativities in your life. So why to become negative? Be positive and start thinking from the perspective of the system. Start talking to the cells, start talking to the protein, start asking them the question. You can start with a very simple question to a plant cell, and you can ask the plant cell that people say that photolysis of water can occur. In the presence of energy of sunlight, water can give you the electrons. But ma'am, please tell me what special you are having, because I am having a swimming pool at my home and the whole day the photo energy is, that swimming pool water is exposed to that photo energy, but they are not giving me current. But what is there inside you? How you are playing with the voltages? So I am having the photolysis of what? Water. Then cell will start giving the answer. And cell will start giving you the answer means you will be searching the right answer. You will be searching the right literature. Right? Now we move to next thing. Again, our developmental pathway. It is not exactly related to cell culture, but it is related to, you can say, overall concept of cell culture. So actually I'm getting an important call. I'm taking a 2 minutes break and then I'm coming back. Just a 2 minutes break. Sorry guys for the interruption. Actually, I got a call, important call. So I'm back and let's continue. So I hope you are liking the things. You are having a different perspective towards biology now and a totally new perspective towards cell division, because I feel it is very, very important to change ourselves, right? Ourselves in terms of we should be modern in every aspect of our life. And being modern means we should be looking at the things from a totally new thing, right? So let's continue. So we are having this presentation again. So I have already explained you that in the normal developmental pathway, we start with embryonic stem cells and then we convert them into progenitors of adult stem cells. Then you can convert them into adult stem cells, and then finally you are having differentiated cells. So on the top of hierarchy, you are having embryonic stem cells, or ES cells, and these embryonic stem cells are having highest differentiation potential as well as highest cell division potential. And you can utilize embryonic stem cells to convert them into any type of cells, and people are trying to uh, use them in different type of cellular therapies. But the problem is, it is very difficult to isolate the embryonic stem cells. Easy from the mouse, but from the human it is very difficult task, and it is unethical in so many aspects. So what people have done, they tried to go back into this pathway. They have taken the differentiated cell and they tried to convert them into embryonic stem cell like cells, and they did the de-differentiation. Okay, and these embryonic stem cells like cells are called as iPSCs, or induced pluripotent stem cells, right? So they are having properties similar to embryonic stem cells. So what is the basic method? You take adult fibroblast cell and you convert them into what? iPSCs, by expressing these proteins which are specific to embryonic stem cells, like KLF4, SOX2, CMYC, Nano, OCT34, Lin28, which I have already explained yesterday. You get the reprogrammed cells, which we are calling as IPS, induced pluripotent stem cells. And these cells can be redifferentiated in the cardiomyocytes, adipocytes, neurons, neural cells, then pancreatic beta cells, hematopoietic progenitor cells and all. So this is one of the specialized cell culture which people are doing. They are doing reprogramming of adult cells into embryonic stem cell like cells, which are called as iPSCs or induced pluripotent stem cells. And they are utilizing these cells for different therapy purpose. Actually, research is going on. Till now, we have not reached to a conclusion or to a stage where we can fully use iPSCs for therapeutic purposes. But yeah, work is going on, and soon there will be some methods, I'm sure about it. Now, one important thing is sometimes we want to start the cell culture with a pure system. Okay. So say I want to grow the CD4 T cells in the culture. But when I take the blood, and from blood I isolate peripheral blood mononuclear cells, PBMCs, then PBMCs are having B cells, NK cells, neutrophil, eosinophil, basophils, monocytes. Then they are having, within T cells, they are having CD4, CD8. So if I start the culture with overall cells, then what will happen? After some time, my cells or my cells of interest, which are CD4 T cells, will be lost. So my culture will be impure and will not be useful. So we can do the sorting of cell. One is by flow cytometry and second is by magnetic cell sorting. In that, what we do, for example, we are putting PBMCs here in the test tube, and I am adding the antibody against the CD4 protein. So these antibodies will be binding all the cells which are expressing CD4 protein on their surface. So in other words, you can say that these antibodies are binding to CD4 T cells. Now these antibodies are tagged with a small magnetic particle. So when you put this test tube in a strong magnet, then what will happen? All the cells of your interest, like CD4, they will stick to the wall because they are labeled with antibodies which is containing small magnetic particles. And cells which are not for your interest, right, they will remain in the supernatant. You can throw them and you can collect your cells. So this method is called as MAX or magnetic cell sorting. So by utilizing magnetic cell sorting, you can purify the population of the cells which you want to grow in the cell culture system, and you can start your cell culture with a purified population of cell. For example, in our lab, we are growing different type of T cells. Then we are growing naive T cells. We are growing dental pulp stem cells. Then we are growing cancer stem cells. So every time we generally and we routinely use this method to purify the population first and then putting the population in the cell culture. Now I am going to explain a complete flow chart of one type of cell culture. So you will be able to understand that whatever I have told you till now, how we are actually applying. And that will be the end of my cell culture discussion. So let's imagine I want to grow the T cells. Okay. So I have to work according to this difficult picture actually. And I will explain you what is that picture. Okay. So even if you are understanding 70 to 80% of this discussion now, that will be more than enough because this is little bit complex. So how I will start the T cell culture? I will take the blood, and from blood I will take the PBMCs, and from PBMCs I will isolate the T cells, specifically purify the T cells with the help of what? Magnetic cell sorting. So now in a tube I am having what? Magnetic cell sorting. Now if I just put them into the media and I wait that they will divide and they will be cultured, their number will increase, then it will never happen. That is why T cell culture is one of the most difficult cell culture system. Why it will never happen? To get the answer of this question, we have to enter inside the body and we have to ask the T cell, ma'am, what do you want? Why are you not dividing in my cell culture? Then the T cell will start giving the answer. Okay, so let's hear in words of T cells. So T cells will say, sir, actually I divide only and only if I get signal to my T cell receptor and my CD3 molecule. And that signal is generally given by an antigen presenting cell which presents an antigen on its MHC molecule. So I will divide. I can start the cell culture. I can make you happy if somehow I am getting the signal of MHC peptide to my CD3 TCR. Okay, then I will tell the T cells, okay, that's fine, I can arrange that thing. Then the T cell is going to say, no, that will not be enough. Other than CD3 TCR signal, I require the signal, the co-stimulatory signal from CD28 molecule, which is given again by two proteins CD80 and 86, which are again present on antigen presenting cells. So if you give me the peptide MHC signal, I will be dividing, but I require this additional signal of CD28, and then I will be able to grow perfectly, grow efficiently, and I will be functional also. Then I will say, that's it, no other requirements. Then the T cell will say, no, wait. If I grow, if I divide, if I become functional, I require the metabolic activation, I require energy, I require so many other survival pathways. For that, I require a cytokine in the environment, and that cytokine is called as IL-2. So there are three essential things for me. Signal from peptide MHC to my CD3 TCR, signal from CD80-86 for my CD28, and for my happiness, for my activation, for my survival, signal from IL-2 cytokine. And these things are generally given to me by what? Antigen presenting cell. So we make a nice bonding with each other. That's our love story. And we call it as a immunological synapse. Synapse similar between two neurons. So if you are fulfilling all these conditions in the cell culture environment, then I will be happily growing. That's great. So I will start culturing what? T cells. Method 1. I can take the T cells which I have purified and I will mix them with antigen presenting cells which are loaded with what? Antigen. Okay, in 1:1 ratio. And if I am doing that, my T cells will get all the signals. I will add some IL-2 in the media for activation and the T cells will be happily growing. Good method, easy to do. But the problem here is I don't know the T cells which I have identified recognizes which antigen, because each T cell is what? Specific. So I don't know which antigenic protein I should add on the antigen presenting cells so they can recognize it and they can divide it. So if I know the antigen, if I know that my T cells are specific to influenza viral protein, then I can add that. If I know that my T cells are specific to this cancer protein, I can add that. If I know they are specific to fungal protein, I can add that. But when I am taking out blood from healthy person, and there are so many types of T cells in a 5 ml blood, because one type of T cell is specific to one type of antigen, then in that case, I don't know which antigen should be added. What I should do? Then I use second method. I remove the APC. I don't use the antigen presenting cell in the culture. Okay, what I do, I tell the T cell, ma'am, you require the signal against CD3. I will add a monoclonal antibody against CD3 in the culture. The antibody will bind to TCR and CD3 and activate them. Okay, ma'am, you require the signal against CD28. I am not having antigen presenting cell, but I can add a monoclonal antibody against CD28. The monoclonal antibody will bind to CD28 and will start the signaling. So the TCR, CD3 and CD28 signal can be taken care of by using monoclonal antibody. And third, what do you want? IL-2. I can add 5 nanogram, 10 nanogram IL-2 according to your requirement. So I am fulfilling all these three conditions by using two methods. One, by using antigen presenting cell if I know the antigen, and if I don't know the antigen, I will be utilizing artificial signals in terms of monoclonal antibodies as well as in terms of IL-2. So this is how normally I can do the T cell culture. Happy? Right? Now, imagine I am having a T cell and in the culture I want to convert them into helper T cell like Th1, Th2, memory helper T cell. So how I do that? Right? Then again I ask the question to a naive T cell that, ma'am, I am giving you CD3 TCR signal by artificial method. I am giving you TCR CD3 CD28 signal by artificial method, and I am giving you IL-2 signal by adding some IL-2 in the culture media. But now you are growing, you are happy, you are increasing your number, but now I want to convert you into different type of what? T cells. How I can do that? Then T cell will again tell, sir, for that you have to read the literature, and the literature tells you this. So this is the naive T cell, which is not specific type of T cell. If they get the signal of IL-12 and IL-18, they convert into helper T cell 1, Th1. If they get the signal IL-4, then they convert into Th2 type of cell. Th1 are specific to virus and cancer, Th2 are specific to parasites and bacteria and all. If you give them cytokine in the culture media, or not naturally, if they get IL-6 and IL-21, they convert into follicular helper T cell, TFH. If they are getting IL-6 plus TGF beta, then they convert into Th17. And if they are getting TGF beta or retinoic acid, they convert into suppressor T cells or regulatory T cells. Now I want to do this kind of system in my lab, in my culture flask. What I will do? I will take naive T cell. First, I will give them the common signal which they require. Monoclonal antibody against CD3 TCR, monoclonal antibody against CD28, and IL-2. These are the basic requirement. Now I want to convert them into Th2. So what I will be doing? I will be adding what? IL-4. After some time, I require Th17 from the initial cells. What I will be giving them? IL-6 and TGF beta. So all these informations we are getting from the cells, and cells are telling us, you trace us, you scan us in the natural environment, you will get the information about the molecules, and finally you will be having the list of the molecules and the list of signals which are required to convert a naive T cell into different type of T cells, and you have to mimic them into what? Your culture flask. So if you give CD3 TCR signal, CD28 signal, IL-2 signal with addition of IL-12 and IL-18, then you will be converting a naive T cell into what? Th1. And this too, Th2 with the help of IL-4, with the help of IL-6 and TGF beta into Th17. So imagine the cell culture. Why I have given you this complex example? Because the cell culture is not about putting 5 grams of sugar, 2 grams of this, DM-EM media, RPMI media. These are the technical parts. But if you are really interested in doing animal cell culture, then imagine for only T cell type culture, you require all these things. And in our body, you are having so many types of cells, multiple type of cells, and every cell is requiring different microenvironment and cell culture media to survive, to grow. And if you really want to master the cell culture, then first you have to learn all these things. What are their basic requirements? And that is the real training of what? Cell culture. The training course or the program of cell culture should not be like this. You go there, okay, how to split them, how to maintain the contamination, how to maintain the sterilization, how to prevent the contamination, how to do trypsin digestion, how to divide them into one flask to two flasks. These things are easy to learn. Cell culture training means getting trained into the real conversion, real maintenance according to their natural environment, according to their niche, learning their requirements, fulfilling those requirements, understanding the health of cells by microscopy, understanding the quality of cells, purity of cells by flow cytometry, purifying them, isolating with the help of magnetic cell sorting or flow cytometry based cell sorting. These are the parts of what? Cell culture. So I hope you liked the cell culture part of discussion. Thank you guys. Thank you very much for your patience. And I hope this discussion will actually help you to think beyond the pipetting and sterilization part of the cell culture, and you will try to learn different type of cells, their requirements, their culture specifications, their culture protocols, and how we can set the new experiments. Reading new books, reference books, and research papers will actually help you with your journey of cell culture. So that's it into the cell culture discussion. Now we will be taking a 5 to 10 minutes break, and then we will be starting the last part of our 2 days webinar, that is the real-time PCR data analysis. Thank you guys. Thank you very much. We meet within 5 to 10 minutes, maximum 10 minutes. I will announce before starting. Thank you. And the questions which you are having from the cell culture, you just please write on a paper or something. I will be giving the answers of as many as questions after the end of the day. Okay, so just have some patience, and then I will be giving the answers definitely after we finishing the second session also. So just take a break of 5-10 minutes, have some water, and be ready for the last part, that is real-time PCR. Welcome back, guys. We'll start in 2 minutes with our final session of this 2 days webinar. I hope you are enjoying the things and getting some new perspective towards the subject. So, we at Logical Biology is having only one aim. We want to tell the people that you can look at the biology from a totally different point of view. And people say that what is the scope in biology, what is the scope in biotechnology, what is the scope in the subject itself, then I always give the answer that scope is not there actually. You have to make the scope. You have to make yourself eligible to get good things. And you can do that by some of the advices which I have given yesterday. Fundamentally correct, giving answers in terms of biology, thinking out of the box, being observer, having a good observation. And as today I said, change your point of view and start asking the questions from the perspective of the system itself. And that is the aim and that is the mission of logical biology, that we want to spread it. So in embryonic stem cell, generally in mouse, Anushri, they kill the pups and they can get some embryonic stem cell. In humans, it is not possible. Few embryonic stem cells you can get at the corner of placenta, but very few. So generally in humans, the iPSC research is booming because it is very difficult to get embryonic stem cell, and actually you cannot have the primary cell culture of embryonic stem cell because in India it is ethically not allowed. You can buy some embryonic stem cell lines also, primary cell culture lines, but that's a different story. So before starting the discussion about real-time PCR, I tell you one very interesting thing. There are so many people who are having the problem with memorizing the things, remembering the things, right? So, according to me, that is my personal observation actually. According to me, memory is our nervous system response, which is the combination of different senses. For example, if you are remembering that that particular color is red, then what do you mean by that? That particular color is red, you are visualizing it, then you are listening it, you are analyzing it, then you are storing the information. That is the memory of your red color. Okay. So if you are having good combination of different type of senses, then you can have permanent memory also. Now, the memory is an interesting thing because people are remembering कि वो जब 5th क्लास में थे तो शिमला गए थे और वहाँ से उन्होंने उस शॉप से आइसक्रीम लेके खाया था. People can remember those things. People never forget the stories of movies. बट हमने 9th क्लास में न्यूटन्स लॉ पढ़ा था, वो अपने को 3 दिन बाद भूल जाते हैं. कल हमने फोटोसिंथेसिस का C3C4 सिस्टम पढ़ा था, वो हम भूल जाते हैं. बट हम स्पोर्ट्स नहीं भूलते हैं, हम मूवी की स्टोरीज नहीं भूलते हैं, हम पुरानी अच्छी जो मेमोरीज है वो नहीं भूलते हैं. सो व्हाट इज द डिफरेंस? Because when you watch a movie, you are listening the movie, you are uh, visualizing the movie, you are analyzing the story, and the same thing you are doing when you are studying. You are visualizing, you are analyzing, you are listening, you are reading. So the number of senses and the number of combinations are similar when you watch a movie or when you read a book. But why we forget the book but still remember the story of movie? I found a reason, and which works for me. Actually, when you are watching a movie or when you were in Shimla when you were 5 years old, at that time you were happy. So there is one more sense, that is the sense of emotion. So with visualization, with reading, with listening, if you add one more sense, that is the sense of happiness, then I feel you never forget that particular thing. And whenever we are reading or whenever we are studying, we are forgetting because the sense of happiness or that extra parameter is absent. And if you start adding that extra parameter while you are studying biology, then you never have to remember it. You can make good memories of biology also. So that's why I always tell my students that whatever you do, you should always be cheerful. If you are cheerful, then you will not forget the concepts. If you are learning, if you are listening a subject, if you are learning science, if you are understanding science, and while understanding and learning, you are having sense of happiness, if you are emotional about it, you are not going to forget it. But you are an emotionless person while studying, you will forget it because you bring out your emotions only while you watch movie. That's why you remember there and you don't remember your books. So whenever we understand the biology, whenever we discuss the biology, we should be so happy, we should be so cheerful that we should get cutis anserina. Now what is cutis anserina? Cutis anserina is the scientific term or the biological term for goosebumps. So we should appreciate, wow, how amazing the hemoglobin is. It is having amazing, it is having amazing 3D structural dynamics that in lungs it is binding to oxygen, having a different 3D structure. In blood it is having different 3D structure. In tissue it is having different 3D structure. You start appreciating it. You start loving the dynamics of hemoglobin structure. You start loving the dynamics of antibody structure. That some of the antibodies are making dimers, some of the antibodies are making polymers. Then you will be having sense of happiness and you will not be forgetting the things. One thing you have to add in your study pattern, study plan, in your timetable, that even if I am studying for 15 minutes, I will be doing with happiness. If for a particular moment I am not happy, I will stop my studies and I will do something else. People say that we study 9 hours, 10 hours. I never believe them because we cannot watch we cannot watch a movie for 10 hours. How we can study for 10 hours? Is it impossible to watch 3 to 4 movies back to back while having same emotions? But we expect our brain will be having same efficiency for 9 hours, 10 hours, understanding a concept without happiness. And people suggest, I mean, people give the interview, मैं 18 घंटे पढ़ता था. I was studying for 18 hours. I mean, come on, 18 hours you cannot watch a movie. You cannot do a sports. You cannot play a game. There is a parameter which is called saturation of neurons also there. So be happy, study hard, and change your perspective. Let's start real-time PCR. And Wilma, welcome. So, Butuan City. In which country is it, Wilma? We're starting in 1.5 minute. Huh? The real-time PCR. So, the next topic which I am going to discuss, that is real-time PCR. Little bit technical. Uh, so we'll try to understand it. I will try to explain it as much as possible, as easier as possible, as simple as possible. And let's enjoy the real-time PCR. So I hope you are able to see this PowerPoint presentation. It is gene expression analysis by real-time PCR. And let's do it. So we know that most of you are aware with the normal PCR, I think. So I will not go into the details of the steps of PCR. So in PCR, what you do, you take a DNA molecule, you denature it at 94 degrees temperature, and then you use two primers, forward primers and reverse primer. Then at 72 degree, you do the extension. You add the enzyme, so enzyme can start from the primers and can make new chains, and then you have multiple copies. You do multiple rounds of this reaction, and ultimately you get millions, billions or trillions of copies by your polymerase chain reaction. So what we are doing in the real-time PCR or the gene expression analysis PCR, what we are doing, we take RNA molecule, then with the RNA molecule we do the reverse transcription by the enzyme reverse transcriptase, and we use a primer which can bind to poly A tail of RNA, and by reverse transcription we create cDNA. Then from this cDNA we start the PCR and we get multiple copies of cDNA. So I can say that if we are getting multiple copies of cDNA, then I was having good amount of RNA previously. So I can indirectly say that the amount of amplified DNA gives us the information about amount of RNA present initially. So if initially more amount of RNA was present, then you are having more cDNA. More cDNA means more amplified DNA, faster amplified DNA. And you can utilize, you can study this information to understand whether a gene is making high amount of RNA, less amount of mRNA, moderate amount of mRNA. So actually here we are going to discuss that how by using real-time PCR we can understand the gene expression. Gene expression means how much mRNA a particular gene is forming, or you can say how much a particular type of gene is expressing in a particular type of cell. So we'll be starting from RNA, then we'll converting that into cDNA, then we'll be doing normal PCR reaction. Now, the conventional PCR, you can detect the amplified product at the end of PCR reaction by doing gel electrophoresis. And by using conventional PCR, it is not possible to calculate what was the initial amount of or initial copy number of template, which is actually important if we want to do the gene expression analysis. That is why conventional PCR cannot give us the information, cannot give us the quantified information that initially how much DNA or how much RNA was present in your sample, which is the basis of gene expression analysis. That's why we shift to real-time PCR. So real-time PCR mein we can detect the progression of PCR reaction at each cycle, and we can use some mathematics, and this real-time detection on that mathematics can help us in quantifying the precise number of initial template, precise number of initial DNA, or precise number of initial mRNA. Right? So I say that there are two types of cells, cell A and cell B. Cell A is expressing more amount of hexokinase gene and cell B is expressing less amount of hexokinase. How you are going to do that? We'll be getting out mRNA, converting into cDNA, doing real-time PCR, some calculations, and you will be saying that you will be giving the information that cell A is making 5 times more amount of hexokinase mRNA than cell B. So this kind of analysis can be done by real-time PCR, where you can detect the amplified product at every step of PCR, as well as you can do some mathematics and can find out the amount of initial template, amount of initial cDNA or RNA. Right? Now, how we are detecting it? Again, very simple. First question is, how do we do the real-time detection? Very simple. We can use a fluorescent molecule which gives us information of increasing amount of DNA by increasing fluorescent signal. How? We can use two types of fluorescent molecules. One can be a DNA binding dye like cyber green, which gives you green fluorescence. Or second, you can use a fluorescent DNA probe. Common term is Ekman Probe. And we can do the accurate calculation of initial copy number. So how we are doing this? Just understand it. So what happens when we do the PCR in a real-time PCR machine? Then we create a graph. The instrument is giving you a software. Software is giving you a graph. And on the x-axis of graph, we are having cycle number of PCR reaction. So we do the PCR reaction for 30 cycle, 35 cycle, 40 cycles, and so on. And on y-axis, we are having fluorescence intensity. So what happens if I am starting with one RNA molecule and converting it into one cDNA, and the cDNA will start giving you copies. So in one cycle, 1 to 2 copy, then in second cycle, 2 to 4, then 4 to 8, 8 to 16, like that. Every time when the DNA increases, the product increases, either your cyber green, which is the DNA binding dye, or your Ekman Probe, which is again fluorescent, bind to this DNA. So with cycle number, the fluorescence is going to increase, because with cycle number, the product DNA is increasing or product nucleotide sequence is increasing. And that is why with increasing increasing cycle number of PCR, you are having increasing the fluorescence. So this kind of graphs. So if you are increasing slowly initially, then suddenly it is going up and then it is reaching to what? Plateau phase. After some time, the fluorescence doesn't increase. This graph is very important. Now, this is the threshold line. Threshold line means this is the value of fluorescence which says that if your PCR reaction is giving more fluorescence than this, then it is significant signal. It is significant signal to do calculation. And if the value is less than this threshold line, then the value of fluorescence is not significant. And we are having a very specific parameter, that is CT value. What do you mean by CT value? CT value is that cycle number where the fluorescence signal is crossing the threshold value. So imagine here, if I put here, then the CT value is coming around 21. So I can say 21 cycle number is the cycle number in this PCR reaction where the fluorescence signal is crossing what? Threshold. Right? And this CT value we utilize to understand the gene expression calculation, to understand the gene expression comparison, to understand different aspects of real-time PCR analysis. Now, CT is that cycle number. It is very important. I will be repeating again and again. CT is that cycle number of PCR reaction where the fluorescence crosses the threshold line, or you can say it is a significantly detected fluorescence. Now, what if the CT value is less? अभी इसको बहुत अच्छे से समझना है. Less CT value means at less cycle number, the fluorescence value has crossed the threshold. Why it has crossed the threshold at less cycle number? Because initially the template DNA or RNA was more, right? So imagine initially if you are starting with 200 RNA molecules, you do the reverse transcription and you get 200 cDNA molecules. And if you do 200 cDNA molecules and start PCR, and the CT value is coming 20. So for 200, the CT value is what? 20. Now if you are starting from only one RNA molecule, so imagine if you are starting from 200, then the reaction will start like this: 200 to 400, then 800, then 1600, then 3200, then 6800. Like this, the DNA amount will increase and the fluorescence will increase quickly. So at less cycle number only, you will be crossing the threshold value. But if you start with one RNA molecule, then it will be 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32. So it will take more cycle numbers to cross the threshold. So if the CT value is low, then initial copy number of the template was high. That's why it crossed the threshold line in earlier cycles. And if CT value is high, then initial copy number of template was low. So it crossed the threshold line in the late cycle. It took time for the system to generate enough fluorescence to cross the threshold. So imagine I am taking a cell A and cell B, and from both of them I am utilizing primer specific to hexokinase gene again. So from cell A, the CT value for hexokinase is coming 15, and from cell B, the CT value is coming 20. So what you will be concluding? You will be saying that from cell A, the CT value is less. It means initially the RNA of the hexokinase was highly present. That's why the initial amount of template was high. The initial copy number of template was high. That's why within few cycles, the fluorescent has crossed what? The threshold line. But in cell B, the CT value is not 15, it is 20, means after 5 cycles, it is crossing the threshold, means the initial amount of RNA or the initial copy number of the template was low. That's why it took time to cross the threshold, and taking time means you are doing the same crossing of threshold after late cycle. So I will be saying cell A is expressing, or the gene expression for hexokinase gene in cell A is higher in the comparison of gene hexokinase in cell B. So less CT value means faster crossing of threshold, means higher expression. High CT value means lower expression. Reason, it is taking time to cross the threshold value, right? So just by looking at the CT value, we can get an idea whether your gene is expressing high or expressing less, or whether the initial template was high or initial template was less. All these things can be done by utilizing the CT value. So CT is nothing but that cycle number of PCR reaction where the fluorescence is crossing the threshold line. Less CT, initial copy number was high, that's why I crossed the threshold line earlier. More CT value means initial copy number was low, that's why I crossed the threshold line in late cycle, right? So people are testing the corona virus by utilizing the real-time PCR. What they do? They take out the nasal swab or any biological sample, they isolate the RNA, they convert the RNA with RNA to cDNA, and they put this primer specific to what? The coronavirus sequences. These primers will bind. With each cycle, the fluorescence will increase and you will be getting the signal. So if the signal is coming, it means you are infected. Signal is not coming, it means initially RNA was not present, and if RNA is not present means you are not infected. So the real-time PCR based kits are using all around the world to detect the corona. Simple principle based on what? Fluorescence, right? Now we just learn how the cyber green is doing that. So initially, for example, you are starting with one DNA molecule. So to this one DNA molecule, the cyber green will bind, and after binding, it will be giving you the green fluorescence. Okay. But with PCR, when the number of product increases, number of copies increases, more and more cyber green is binding to the product and the amount of fluorescence is increasing. Similarly, so you can see that. So initially the binding of cyber green is less, then slowly increasing, crossing the threshold line and suddenly going up. Similarly, the Ekman probe is working. So Ekman probe is the specific sequence against your target DNA sequence. It binds to the target sequence, and if the extension is happening, then it is partially displaced from its position, and the reporter start giving you the signal. So even if you are not going into the complexity of Ekman probe, you just learned that it's this more specific method because it is binding to your target sequence, and if the amount of DNA is increasing, the binding will increase and obviously the fluorescence will increase, and the similar graph you will be getting. Initial binding is less, and after that binding is increasing, crossing threshold, and ultimately reaching to what? Plateau phase. So till now what we have understood, that we are measuring the fluorescence, and from the fluorescence threshold we are measuring the CT value, and CT value is giving us the information about the expression, whether the initial amount was high or low. How we do that? The experimentation. We take the cells, isolate the RNA, we prepare the cDNA out of RNA, we do the real-time PCR with cyber green or Ekman probe, we obtain the CT values, then we do the data analysis. That's it, right? CT value is low, the initial copy number of template was high, it means amount of mRNA of gene was high, means gene expression is high. CT value is high, means initial copy number of template was low, means amount of mRNA of gene was low, means gene expression is low. More CT, less gene expression. Less CT, more gene expression. That is what we do. But we are having a problem. That is the most complex part of the real-time PCR analysis. It is looking a simple flow chart. We are saying CT low, CT high, CT low, high expression, CT high, less expression. But that is not completely true. Reason. You have taken two samples, two types of cells, sample 1, sample 2. And you are doing real-time PCR for gene X, right? Then for sample A, the CT is coming 20 for gene X. And for the sample B, CT is coming what? 22. By looking at this, you will be saying that, okay, CT value is less. It means the expression was gene X was high in sample 1 because it crossed the threshold of fluorescence earlier. And if the sample B is having CT value 22, so you will be saying sample 2 or sample B is having less expression of gene X because it took time, it took more cycles to cross the threshold value. But it is not correct. Reason. You will be making this conclusion that gene X is expressing more in sample 1 or sample A than in sample 2 or sample B. But ऐसा होता नहीं. What is the problem? What if initially sample 1 was having higher number of cell than sample 2? So it is possible that by mistake, here you take 10,000 cells and you take 9,000 cells. So 1,000 cells are extra. So you are getting extra RNA for gene X. And if you are having extra RNA, then because of your technical mistake, the experiment become a biased experiment. The CT value which is coming less for sample A is not because of higher amount of RNA or higher gene expression, but because your sample A or sample 1 was having higher number of cells. That is the biggest problem in the concept of real-time PCR, that humans can do the mistake, and the mistake is they can take different number of cells, and it is impossible to take identical number of cells. You cannot have a method by which you get 1000 cells here, 1000 cells here. Even if there is difference of 5-10 cells, there can be biased results. Now how we solve this problem? We solve the problem by normalizing this CT values or the data with the reference gene. And we use a method which is called as 2 to the power minus delta delta CT method. I will be explaining how we are going to do that. Now, what is the reference genes? Reference genes are those genes which have nearly same expression in all cell types, like genes of glycolysis, that is GAPDH, gene of beta actin, that is cytoskeleton, gene of RPS, that is ribosome. What happens if you are having 10,000 cells here and if you are having 8,000 here, then you will be having different CT values for gene X, 20 and 22. And this difference is not coming because your gene expression is high in case A, but it is coming because sample 1 was having higher number of cells. But if you do the real time for GAPDH also, then you will say, okay, in GAPDH also the CT values are less in A and high in B. But logically the GAPDH expression should be equal in sample A as well as sample B, because these are housekeeping or reference genes. But even in the case of GAPDH, if the CT values are different, it means you will say we have done a mistake, we have taken a different cell number, right? Because what will happen if your cell number is same, like 5000 and 5000, then the gene expression of X can be different, but the gene expression of cytoskeleton cannot be different. So for gene X it can come 20 and 22, but for beta actin it should come 16 16 or 15 15. Getting my point? Or similar. So it is coming different from gene X as well as different for beta actin also. Then you will be saying, okay, we did a mistake, we have taken different number of cells. That's why we are doing real-time PCR of the target gene as well as we are doing real-time PCR of the reference genes. We get the CT values from the target as well as we get the CT values of what? Beta or GAPDH or RPS. Then we normalize the data. Now the mathematics will come and I will teach you how we are normalizing the data, and we are not only saying the expression is high or low, but we are saying how much high or how much low we can quantify it. So we'll be doing that analysis in a very step-by-step easy method. 2 to the power minus delta delta CT method for relative quantification of gene expression by real-time PCR. Now, we are doing this experiment. We are having 1000 cancerous cells and 1000 normal cells and want to check the gene expression difference in these two types of cells for which gene P53 by real-time PCR. That is our target. Okay. So what we are going to do, we will take 1000 cancer cells and 1000 normal cells. Theoretically, practically you cannot take. The difference will be there. Maybe it will be 1200,000 here, maybe 1100,000, maybe 950,000. But we want to check the expression and we want to say whether cancer cells are expressing P53 high or normal cells are expressing P53 high and all. How we are going to do that? Real-time PCR we will be doing. From both the samples we will take the cells, we'll isolate the RNA, we'll do the cDNA synthesis, we'll do real-time PCR either with CyberGreen or either with Ekman Probe, and then we'll be obtaining CT values. But now we will be doing cDNA and real-time PCR for both, for P53 as well as for any reference gene like GAPDH, beta actin or RPS and all. Okay, and we are getting these CT values. So these are the normal cells. The CT values are for P53 15 and for cancer cells 12. So if I don't look at the CT value of GAPDH or the reference value, then I say that normal cells are expressing less actually because they are having higher CT value, and cancerous cells are expressing high because they are having less CT value. But this is not completely true because we have to take the reference gene into the consideration. So the reference gene CTs are 16.5 and 15.9. Okay, so if you are looking here, then conclusion is what? P53 is expressing less in normal because high CT value, and high in cancerous cell because of less CT value. But let's do the calculation. Let's normalize the data and then we'll be seeing. Right, so how we'll be normalizing the data? So these are the CT values for P53, 15 and 12 for normal and cancerous, and for GAPDH 16.5 and 15.9. First, we will be doing delta CT. So what is delta CT? Delta CT for each sample will be target gene minus reference gene. So the target gene is P53 and reference gene is GAPDH. So that is 15 minus 16.5 is equal to 1.5 minus 1.5. Then we can calculate the delta CT for cancerous sample. How? Again target gene minus reference gene. So the target gene is 12, you can see here, and the reference gene is 15.9. So the difference is minus 3.9. So we have calculated delta CT normal and delta CT cancerous. Then in next we do the delta CT. So what is delta CT? Now cancerous minus normal. Okay, so the cancerous delta CT was minus 3.9 and the normal was minus 1.5, and we are getting a value. Then we calculate 2 to the power of these values. Okay, and we are getting a value, for example 5.3. So now what this 5.3 is indicating? This 5.3 is indicating fold difference of gene expression. It means the conclusion is in cancer cells the P53 gene is expressing 5.3 times more than normal cells. Okay, and how? Again, first CT target minus reference from both the samples. Then delta CT cancerous minus normal, or you can do normal minus cancerous also, but this statement you will be changing, which is more than normal or which is less than normal. And finally, whatever value are you getting, you do the 2 to the power. Why we are doing 2 to the power of this delta CT? Because in each cycle the product is getting double. 2 to the power 1, 2 to the power 2, 2 to the power 3, 2 to the power 4. That is how it is happening. Okay, so delta CT is always target minus reference, and delta CT is sample minus control. So in this case our sample was cancerous and the control was our normal cells, and 2 to the power minus delta CT is actually fold difference of what? Gene expression. Let's do one more experiment. Okay, so now we are looking for to analyze the gene expression of an oncogene X after 3 types of treatments on the cancer cells. Okay, so we are having 4 samples: control, untreated cells, then treatment 1, treatment 2 and treatment 3. For gene X we will be doing real-time PCR again with the same method, and we'll be calculating CT values 30, 27, 26, 25. If I look at this table, then I will be saying in control it is expressing very less because CT is high. Then in treatment 1 it is expressing high because CT is decreasing, so expression is increasing according to only CT values. But that is not true. We have to normalize the data. So we do the CT of GAPDH also, and we are getting. So for each sample we are having CT of gene X and CT value of what? GAPDH. What we do? Again CT of gene X, CT of GAPDH. Then for each sample we do delta CT, target minus reference, right? Then we calculate delta CT, that is treatment minus control. So here the delta delta CT for the control will be what? This minus this 0, then treatment minus control, then second treatment minus control, then third treatment minus control. Okay, whatever values are coming, you do the 2 to the power minus delta delta CT and you get what? Fold difference 1 4 16 16. So you will be saying your gene X is expressing one time in your control, but with first treatment it has increased 4 times, with second treatment it has increased 16 times, and in third it is increased what? 16 times. So that is how you make the graph of relative gene expression, and in control the gene is expressing one time, in treatment 1 it is expressing 4 times, then 16 times, and then 16 times. If you are having multiple samples, you can utilize this method in an Excel file, and you can easily calculate the delta CT, then delta delta CT, and then 2 to the power minus delta delta CT, which is actually equal to fold difference of the gene expression. So guys, that's what that is it from my side in the real-time PCR analysis. So thank you very much to Patkar and Thakur College for organizing this beautiful webinar. Thank you very much to all of you guys that you have shown your great patience with me. You listened to me, and the crowd was very good, responsive, and it was an amazing experience. So I hope you liked all 3 sessions. So I will tell you about ourselves. We are Logical Biology. We are having an agenda that providing online lectures, online video courses to the students so they can learn the biology in the most logical and interesting way possible. We are having so many online live courses already running, that is Fundamentals of Advanced Biology, Flow Cytometry, Epithelial Mesenchymal Translation, Cancer Biology, Photosynthesis, Respiratory Biology. We organize so many webinars, how to study some subject, like how to study genetics, how to study cell biology, how to study biochemistry. Then we are having some upcoming courses on NMR spectroscopy, cell signaling, 3D protein structure, advanced course, a very long course on cancer biology, a very long course on immunology fundamentals and advanced. We are also taking live classes as I am taking this webinar for different masters and PhD exams like CSIR, GATE, IIT, JAM, TIFR on every weekend, Saturday, Sunday. And we are focusing how people should crack the interviews and the group discussions. We are making comics, amazing comics, so you can understand the biological concepts in easier way. If you are interested in our programs, joining our courses or on our online live classes, you can write to us at logicalbiology84@gmail.com, or you can WhatsApp us at this number 911-366399. Then we are having our lab also, where we do research collaboration in the field of cancer biology and stem cell biology. We even provide the training for the faculties and professors in the advanced technology platforms. Then we are having college and university collaboration for online and offline courses. We organize seminars and webinars. We do many scientific events. We have created comics till now on opsonization, love story of T cell, virus infection, bacteriophage, common cold. We help in paper writing, SOP writings, and so many other things. So thank you guys. Thank you very much. If you want to contact to me directly, you can write to me at logicalbiology84@gmail.com, or you can WhatsApp me at 86986844792. And if you scan this, then you can follow us on our YouTube channel, that is Logical Biology, our Instagram, our Facebook. So thank you guys. Thank you very much for your patience and for your presence. I am ready to have some questions on cell culture as well as on real-time PCR. Smita, you have asked me this question yesterday also. So it is very easy method that are you actually interested in doing cancer research or not, there is a book called as Fundamentals of Cancer Biology or Biology of Cancer, I think. The name of the author is uh, Robert Weinberg. Okay. So if you are really interested in making your career in cancer research, then you start with that book. That is my advice to every person who is interested in cancer research, that start with that book, read all the chapters, and then join a lab to learn the technology like apoptosis analysis, cancer cell culture, and all these things. So these are two steps which are very important. One is reading that book, and second is understanding or learning some techniques, and then you will actually get to know whether you are made for the cancer research or not. Okay, and if you want any kind of help in the complete system, then you can contact me. You can WhatsApp me on my number, which I have typed here, 869868479. I am having one question. Anand sir, I am coming up with a video within 3 days on my YouTube channel on CAR T cells. So it will be detailed analysis of CAR T cells, how we prepare them, what are the experimental protocols, and what are the applications. So you can go through that 1 hour video and you will be having all the relevant informations about the CAR T cells. Anushri, ESC also have specific? Yes, we are having some markers, some proteins which are embryonic stem cell specific, but generally we prove that something is embryonic stem cell, some cells are embryonic stem cells by doing real-time PCR for their specific transcription factors. Okay, so we do the real-time PCR for OCT4, NANOG, and c-MYC, KLF4. If the cells are expressing these markers or transcription factors, they are giving the signals in real time, then only we are sure that these are embryonic stem cells. So generally for embryonic stem cells, both flow cytometry and real-time PCR is done. So Chitra, I mean, yes, what is happening in the field of circulatory miRNA, you cannot take the normal reference genes because of the size difference. But I can provide you a link where the Thermo Fisher has come with specific stem loop sequences which can be utilized for the normalization of data in case you are doing the gene expression analysis or the expression analysis of circulatory miRNA. So I have checked personally those sequences for my experimentation and I am getting good result because we have developed a protocol in our lab where we are doing microRNA expression analysis by flow cytometry also. So we do real-time PCR as well as flow cytometry both, and when I compared the data sets, then these stem cell loops are working amazingly. So if you want, you just say hi to me on WhatsApp and I can give you the link. Okay, yeah, Yamanaka factors. So our lab is situated in Pune. It is Nirav Biosolutions Private Limited. So that depends upon every decision actually, Uzma, every decision taken by a particular cell type depends upon the external signal. If you are not having any requirement for the repair, so you don't want to get differentiation. So for example, how the HSCs decide that they have to create new blood cells. They get the signal in terms of either hypoxia or in terms of any external injury where you are losing the blood. And when it is happening, then obviously the HSCs get the signal. Now they have to do asymmetric cell division and have to create new blood cells. So in normal condition, those injury signals and those hypoxic signals or those repair signals are absent. So they go for symmetric cell division. So cell signaling I like personally as a branch of biology, because there you actually learn that how different type of decisions are taken by cells in terms of division, repair, differentiation. So generally when there is requirement of replacing the old cells, then you go with asymmetric cell division. Yes, if we are going for practice problems in genetics, I mean, if you want to do some question practice, then I prefer the iGenetics by Peter Russell. And if you are interested in reading the theory part, the concept part of genetics, then you can go with the Genetics by Griffith. It's a nice book. I personally use that book to read theory or read concept sometimes. But for the question practice, I use iGenetics, Peter Russell. Yes, we can trace expression of two genes with single PCR experiment. It is very simple. You have to use two Ekman probes, because Ekman probes are specific to a particular sequence. You can go up to 5 also. And I think now with new machines, you can go up to 7, 8 also. So in our lab, we are having Quant Studio 5, where we can use 5 types of Ekman probes simultaneously. It means we can analyze 5 types of genes simultaneously. Yeah, obviously, if your primers are non-specific, then how you are going to be sure that the products which are giving coming out, or the signals which are coming out, they are coming from your gene only, right? So we don't isolate single type of RNA, right? We isolate all the mRNA from the cells. So it is very important to select a very specific primer. And primer specificity is generally initially screened by the bioinformatics, and then for the GC content, then melting point, and many parameters are there to select specific primer, removing the non-specificity. So primer specificity is one of the most important point in terms of setting up a PCR experiment, or more importantly in real-time PCR, where you are isolating multiple. Cancer, it is the Biology of Cancer by Robert Weinberg. So generally what happens when you read cancer from a normal cell biology book, it's one chapter. So in Biology of Cancer by Robert Weinberg, oncogene is one chapter, tumor suppressor gene is one chapter, metastasis is one chapter, angiogenesis is one chapter. It's a 1500 pages ka book, I think, where each topic is having one chapter specific designated to this. No, so 5 colors in real-time PCR, the dyes which are available right now, which are we by which we are tagging the probes like Tamra and all. So they are having good spectral reasons where the overlapping is very less. So up to 5, actually you can select 5 dyes which are not having at all the spectral overlap. Uh, Generally, the reference genes are expressing more than the target genes, because these are housekeeping genes and they express in higher amount. So obviously the cells will be mainly focusing on the metabolism, ribosome synthesis, cytoskeleton. So we get less values of uh, reference gene in comparison to the target gene. Less value means they are expressing high. And the reason is simple. Cells are focused to, these are the housekeeping, these are the most important genes which they have to continuously produce. So the specific genes are dependent when you want to produce them, when you want to express them, whether you are having external signal to express them. So we generally get lesser value of CT uh, for the reference genes in comparison to target genes. It's a general condition. So guys, any other question? So I think that's it for today. So once again, thank you very much, guys. And if you want to become part of our WhatsApp groups, where we time to time organize different types of webinars and all, you can contact me on my WhatsApp number. That is this, and we can have some very good biological discussions on different topics. So please like and share our videos, subscribe to our channels, and if you're having any specific query, you can always contact us. Thank you guys. Bye bye. All the best for your future endeavors. All the best for your study plans, research plans, whatever you are having. And I always suggest make some change in your perspective and do amazing things on your life. Thank you guys. Thank you very much. Bye bye.